I am not a professional genealogist or a certified genetic genealogist. My undergraduate training was in geology, not genealogy, and my graduate work was in clastic sedimentology. And I worked as a physical oceanographer, geophysicist, hydrographer, and this is about as far from genealogy and biochemistry as you can get. Until in the early 2000s, I was aware of genetic genealogy, but I had no interest in it because my paper trail is so extensive. What was it going to teach me? What was I going to learn from genetic genealogy? And what happened was in 2018, some of my wife's cousins started getting ancestry DNA tests. And they essentially cajoled or goaded her into getting an ancestry DNA test. And I said, what are you going to do that? Why do you want to do that? Well, she just did it. And that sort of prompted me to get a DNA test, except I, being aware of the technology and a little more conversant with it than other people, I didn't go to Ancestry to get an autosomal DNA test. I went to Family Tree DNA, got an autosomal DNA test, a Y chromosome test, and a mitochondrial DNA test, which we're going to discuss during this talk. And I essentially, even though it didn't reveal, I was correct, it didn't reveal too much that was terribly interesting. I got interested in the subject, and I have tested now with Family Tree DNA, Ancestry, and 23andMe, and I have uploaded my DNA results to other services, and I'm, getting matches around the world and that sort of thing. About my talk, originally it was intended to be an update to the Sons of the American Revolution chapter on the changes in the acceptance of DNA data for applications to SAR membership. But after that 5 minutes was over, what else was I going to talk about for 25 minutes? So I expanded the talk, and it grew. And so now we have the talk that I have now, which is considerably longer than 5 minutes. There are 23 slides. If I talk a minute and a half for each slide, we'll be here 30 minutes. If we talk 3 minutes for each slide, we'll be here an hour. If we talk 6 minutes for each slide, we'll be here for 2 hours. It's up to you. And I will be mentioning my Newcomb genealogy frequently because it is important in certain aspects of the DNA testing, and some things that you might want to know if you have your DNA tested and you start looking into the results and trying to interpret the results. Let's see if this works. All right, a little history of DNA science. In the 1800s, Gregor Mendel started this field of empirical genetics, where you may remember from junior high school, his experiments with fruit flies, where you had red eye and green eye fruit flies, and you bred them in the various characteristics passed on, and he began understanding how the male and female contributed to parts of the genetic material. Through the 1800s, as microscopes improved, people began to realize there were these little things called genes in this nucleus of the cell that did stuff, but they didn't understand what they did. They could watch them, but they didn't understand In the 1940s, there was beginnings of research into genes and the interrelationship of genes and proteins. But the big breakthrough came in 1953 when Watson and Crick discovered the DNA's double helix structure. In the 1960s, when I was in junior high school and high school, there was research into how replication, meiosis and mitosis worked. And by 1984, they started the Human Genome Project, which completed in 2003 with complete mapping of the human genome. In 2000, Family Tree DNA initiated consumer DNA testing, and today you can get full genome sequencing on a single test, a single chip. And when I talk about chips, there are these glass or plastic plates that are etched that contain tiny microscopic cells, and in the cells are sequences of RNA that attract similar sequences from your DNA sample. DNA is deoxyribonucleic acid, RNA is ribonucleic acid, and it's a double helix that's linked through these coding material, which are cytosine, guanine, adenine, and thymine, and they form the letters C, T, G, C, G, A, and T, which are the alphabet that the genes are made of. And some of this you may remember from high school biology. How these chips that they use are just amazing, and I don't claim to understand how they work, except they replicate your DNA and then they expose it to these chips, which glom on to certain parts of the DNA and that match them, and only that match them. And from that, they are able to determine what's called a single nucleotide polymorphism, or SNP, and this is what is measured or recorded in DNA testing. So in my lifetime, from the early 1950s, we've gone from knowing the genes were some sort of protein to being able to completely determine your human genome on a single test. Now, here are some uses for DNA data: medical, law enforcement, paternity determination, forensics, husbandry, and genealogy. And I'll discuss the husbandry right now because I don't have a slide on that. You can get your dog, your cat, your horse, your bull, your goat. You get them all. There are DNA tests for them to determine what breed they are and maybe whether or not they have any genetic predisposition for illnesses. So it's not just limited to human. Okay, medical. Some diseases have strong DNA links. Some breast and ovarian cancers are strongly linked to what's known as a BRCA1 and BRCA2 gene mutations. There is a disease called the von Hippel-Lindau disease, which is a whole series of debilitating maladies that are genetically linked. Down syndrome and cystic fibrosis both have a strong genetic link. If you've ever had a PCR test, have you had them shove a Q-tip up your nose till they hit the back of your brain and get a sample for COVID and send it off? What they were doing is they were taking a sample of the virus to check its RNA or its genetic material through a thing called polymerase chain reaction, where they take a very small sample of a DNA or RNA and they run it through cycles of temperature up and down fairly rapidly, and every time they run the cycle up and down, it doubles the amount of genetic material that it replicates. So it's 2, 4, 8, 16, 32, 64, etc., until it gets up to thousands of times. And then they can test that material for whether or not it contains a specific genetic material of the virus that they're looking for. mRNA vaccines. You remember Bones here from Star Trek, and how in 30 minutes with 5 commercial interruptions, he could cure any disease in the world. Well, in 2010, Moderna was founded, and in 2008, BioNTech was founded for the express purpose of rapid development of mRNA, messenger RNA vaccines based on analysis of DNA. And people who are vaccine skeptics say it took Salk and Sabin years to develop a polio vaccine. How could anybody develop a vaccine for COVID in a month? And then the longest part of the process was figuring out how to ramp up production and doing the tests. Well, the reason is that the companies that did this were specifically formed to be able to respond rapidly to new developments, new viruses, and that sort of thing. So we're in the era where Bones comes up with cures through DNA analysis in 30 minutes. And Medicare fraud. Some unscrupulous doctors found out that they could charge Medicare $5,000 each for DNA tests. They sent out recruiters to senior centers and groups like this saying, Medicare wants you to get tested for, your DNA tested for diseases, for inherited diseases and things like this, and we'll take the sample, spit in this cup, and so forth, and it won't cost you anything. $2.1 billion is estimated to have left the Medicare Trust Fund before this scam was shut down. Some of these diseases, some of these genetically linked diseases are bound by the DNA. It is tightly wound up, and then those parts of the DNA are not active because they're bound. And it takes some activities such as stress or illness or something like that to trigger the disease taking place. And that's beyond the scope of this talk, but I just want to point out that there are medical uses for DNA testing. Law enforcement, some arrests and conviction require submission of DNA samples for inclusion in a nationwide database. Crimes have been solved by expert analysis of subjects' DNA comparing with public DNA databases, particularly one called GEDmatch. And there's the forensic identification of remains. It used to be when someone went missing, they said, do you have their dental charts? Now they want to know is, do you have their hairbrush or toothbrush? And they will collect a DNA sample and keep that on file in case they find a body. Paternity and adoptees. DNA is commonly used to prove or disprove paternity, obviously. Used by adopted children to locate their biological family. And this is one that is interesting. It's used by people to track down their sperm or egg donor. And I have run into people online who are trying to find their father, the sperm donor, so they could sue him for their genetic disease. Probably on the theory of wrongful birth. And I don't know how that would ever come out, but I would hate to be the one that had to defend myself against such a claim. All right, now we get to genealogy. There's only a small fraction of your DNA is actually analyzed, as over 90% of your DNA doesn't change very much and is used to control basic functions such as cell division, and only a small percentage of it is different from an ape. There are 62 million base pairs that make up the human genome, and only about 100,000 of them are normally tested for autosomal DNA and an autosomal DNA. DNA also has what's called non-coding sections, which are sections that they haven't figured out that they do anything, that they don't replicate proteins or do anything, that they're just linkages. Genetic genealogy opens a lot of doors, and some of them are closets. My wife's cousins sort of discovered they're part African American. They have sub-Saharan African heritage. They're from New Orleans, so Granny still hasn't quite accepted it. The autosomal DNA refers to the 22 chromosomes 1 through 22, plus the X chromosome. And these are, and it's all the chromosomes except your mitochondrial and Y chromosome. And they look for single nucleotide polymorphisms, or SNPs. And it's used for ethnicity estimates, matching genetic cousins. And this DNA is a product of mixing both parents' genes. It runs from $39 to $100. And I'm going to discuss what's called an X chromosome match. By the way, if anybody has any questions, just raise your hand. We've got lots of time, and we can expand on this. Like I told you, I can spend 6 minutes per slide, and we'll be here for 2 hours. One of the complaints I had of some of the times I've given this in the talk in the past is that they said, well, it's a 30-minute. You're kind of limited to 30 minutes. And I said, well, we have to rush. So, okay, mitochondrial DNA, both males and females, inherited unchanged from the mother. And it usually, the mitochondrial DNA is very small. It's a ring of about 100,000 base pairs. And it's usually analyzed completely, and it runs about $130 for the analysis. And then the male's Y chromosome, only males have Y chromosome. It is inherited unchanged from the father, and you are first put into a haplotype, which is based on the single nucleotide polymorphisms in the Y chromosome. And then within that haplotype, you're differentiated by what's known as short tandem repeats, and they are like the DNA is stuttering. There are a varying number of stutters in the DNA, and how often and how frequently different locations in the Y chromosome stutter with short tandem repeats is a differentiating factor within a haplotype. So these are the STRs at specific locations. Analyses range from what's called Y12, where they check 12 locations for STRs, to Y700, and it costs between $100 and $400 to do this test. Okay, autosomal DNA. All right, this is what most people do when they say they get a DNA test. All right, and DNA analysis looks at chromosomes 1 through 22 plus the X chromosome, and it gives you a guess as to the Y haplogroup. These chromosomes got to go meiosis, which is the mixing of the male and female components, and the DNA gets scrambled, rearranged, recombined to make something new, all right, between the father's and mother's DNA. It's based on SNPs. It's used in ethnicity estimates and locating genetic relatives. We'll talk about Y chromosome matches. And it is important that you understand that on every time you do a DNA test, there are things called no-calls. When they go to read these little cells in the DNA chip, it's supposed to be either this or that. And sometimes they can't determine whether it's this or that, and that's called a no-call. And when an SNP has a no-call, then it can't be used in an analysis. And we'll get to this a little bit. Morgans or centimorgans, or CMs, are the units of DNA that is shared between two different people. Okay. And the no-calls, they say DNA tests aren't accurate because we had identical twins who did DNA tests and they came out with different ethnicities. That's because the ethnicity estimates are so sensitive that a few no-calls will throw them off. And so two people or one person can do DNA tests on the same day and come up with slightly different results. Two identical twins or the same person. And testing, you can do tests with different companies and get different results. They use different chips, different technology. Okay, ethnicity estimates. This is the reason that 90% of people do DNA tests. And it is about the worst possible reason to do a DNA test. It is the least accurate, and here I have a what's called a tin BB puzzle. You may remember these from your childhood, where you've got this little cup that has a glass cover and BBs, and you try to roll the BBs around, and if you hiccup, the BBs all go sliding off to one side or another. That is ethnicity estimates. If they get you to the right continent, they're doing good. Do not get upset because a ethnicity estimate says that your ancestry has Hungarian and you're Austrian. You know, it's not, there's not a change there. The DNA doesn't follow national boundaries, and these estimates are so sensitive to slight changes that they can't be relied on for anything other than just conversation at a party. Do not get divorced or disown your parents or anything like that because of this. And some ethnicities are particularly difficult to pin down. German is one of them. Every invasion in the world passed through Germany. Okay, so now we're going to get into my DNA results. Okay, and this is per ancestry. Like most Americans, I'm Heinz 57 variety. My mother's ancestry was, her parents were right off the boat from Sweden and Germany. My father's ancestry partially goes back to the Pilgrims. And that's reflected in my DNA results. You see Sweden, Germany, Scotland, England, Ireland, Portugal. Where did Portugal come from? Yeah, I have no ancestry in Portugal. One of the things that's interesting is that we don't know where in Ireland my great-grandmother came from. We know a cousin of hers was born in the village or the parish of Mulla in County Cavan. Ancestry shows this bullseye in northeastern Ireland, right over County Cavan, and right over the parish of Mulla. And how that got there, how my DNA is specific to that spot, I have no idea. On the other hand, here is my family tree DNA. Here is Scandinavia, England, Ireland, France, Germany, Poland, Finland. I don't know about Finland, you know, it's close to Sweden, but Greece, Hungary, you know, it's not something you get upset over. Different companies have different ethnicity estimates. You don't worry about this. Other sites, which I've sent my DNA to, give different results. 23andMe just has countries, say broadly, Central Europe, broadly, England and Ireland, and so they don't, 23andMe doesn't try to be specific to specific areas. What autosomal DNA is very valuable for is genetic relationships, finding your cousins. So autosomal DNA can be used to find genetic cousins, some of which may come as a surprise. You can locate out to about 6 cousins. But it will only tell you which side of the family, which side of your mother or father's side of the family, with an estimate of the closeness of the relationship. 6 cousins is about 10 centimorgans, and we're going to get into centimorgans in a little bit. It is important to understand when you think about meiosis and the mixing of the father and mother's DNA and the recombining of the DNA, it is a fruit salad. It is not a fruit smoothie. It doesn't get run through a blender and then stuck back together again. There are hunks and chunks that break apart and get recombined. And that is important in some of the advanced analysis of autosomal DNA. And you may not have any of your great-grandmother's DNA. Actually, you may have some of your great-grandmother's DNA, but it's just not part of what's analyzed in the 100,000 points that are analyzed in a genetic DNA test. And it may not be different. It may be her DNA, but it may not be different from anybody else's DNA. We're going to discuss what's called the Leeds method and X chromosome matches. And if you have access to the complete DNA map of an individual and to a group of individuals who are related, then you find that the DNA segments overlap. And you can find people that you are related to by linking these DNA segments together and building a more complete picture of an ancestor's DNA or the matches for your DNA. There is, in fact, a procedure called the Lazarus technique, where if you have enough samples of people that you match, you can reconstruct part of your ancestor's DNA signature, not the actual DNA, but what their DNA would look like, and find people that are related to them. We don't have to spend a lot of time. Centimorgans are a unit of measurement describing how closely 2 people are related. Identical twins share 7,050 centimorgans. A parent and child is approximately 25 centimorgans, half of that of the identical twins. Half cousins are approximately 440 centimorgans. So this is how, now the formula for calculating centimorgans is this mess right here. It's defined as the distance between chromosome positions, foci or markers, which are expected to average number of intervening chromosomal crossovers in a single generation is 0.01. And I can't explain that. I can just tell you this is what people use to determine how closely related you are to someone else that you match. And different testing services have different databases, have different ways of calculating centimorgans. So if you're related to you have a cousin who is in family tree DNA and in ancestry DNA, they will show different numbers of centimorgans. Okay. The leads method, one of the things you can do is find out people that you are related to in common with other people. And the leads method takes and groups the people who are related to each other and to you, and they put them on a matrix. And descendants from a different ancestor group together in these blobs. And this is an automated leads method spreadsheet type of affair. And clusters can be assigned to individual ancestors, and it's also a tool for testing endogamy. Anyone need endogamy? It is married cousins. Okay, and all right, here is where we need to discuss some aspects of my genealogy. And that is when my wife talked about getting her DNA tested, I said, why would I want to get my DNA tested? I've been doing genealogy for 40 years. Well, at the time it was, 35 years. This is the Newcomb 1923 genealogy. This is the 1874 genealogy. My family has been doing genealogy for 150 years. Do you know how hard it is to get cousins to do DNA tests? When they've got a genealogy they use as a booster seat for their grandkids at Thanksgiving. I have been trying to get my only living male first cousin, my second cousin, and my fourth cousins to do a DNA test to help me out with the problem. And they refuse. They say either I'm afraid of the privacy problems, or what is it going to show me? I've got this genealogy. What good is it going to do me? So you might think the colonial, family Newcomb from the 1600s is going to be up here at the top. These are the Campbells. They are a little researched Scotch-Irish family from Franklin County, Pennsylvania. And they show up on the top of the list because they do a lot of DNA testing. And they put their DNA on different servers and things like that. This yellow blob here is what I call the Waters-Hickok vortex, which is a family that I am somehow related to, that I can't figure out how I'm related to them, but the whole bunch of them have tested their DNA and have scattered it on all the different matching sites and uploaded it, so they're on all DNA sites. So it's the people with the questions about their genealogy that get serious about DNA testing, and the Newcombs are down here somewhere. The biggest family, all my German second cousins seem to have tested. A lot of my Swedish cousins have tested. The Newcombs don't test. Okay. We'll get back to this subject because this is important when you're analyzing your DNA results. If you're going to do more than just determine whether or not you need to buy a kilt or lederhosen. The X chromosome is everybody has an X chromosome, females have 2, males have 1. The male inherits the X chromosome directly from his mother. The female X chromosome undergo meiosis, where they mix between the mother and father. But it's inherited in this particular pattern, and one of the bits of information that the testing services give you is an X match, yes or no. And if you are an X match, this is a male, here is soon to be a male here in this spot. His mother, his father, or his grandfather and his grandmother. So the male inherits the X chromosome from his mother. The mother inherits the X chromosomes from both parents. And so forth and so on, going back out through time. So if you are an X match, you have to fall in one of these boxes. Of course, if you start off as a female, you just start at this point. If you're a male, you start at this point. So it's a good detective tool for telling if someone is an X match, they are going to be on your mother's side, if you're a male. And so it's important to understand when you're doing an analysis of your DNA matches, whether or not someone is an X match. I don't have a lot of X matches. It hasn't been important in my research. Okay, the mitochondrial and the mitochondrial DNA. They are bodies inside the cells that have their own DNA. They facilitate energy production in the cells, and they may have originated as separate organisms such as a virus, which are distant ancestors, and by distant ancestors I don't mean monkeys, I mean plants. Found useful and decided to adopt. They are inherited from your mother, and their DNA changes only very slowly, and it does not undergo meiosis. There are approximately 100,000 base pairs, and the DNA is in a ring. And whereas the DNA, the chromosomes in the human, the other chromosomes are Y-shaped with a link in the middle, or not Y-shaped, they're X-shaped, except that they get more coiled up. And so males and females both have mitochondrial DNA, and it can be analyzed. And here is the results of my mitochondrial DNA analysis. This is a map of Europe, and all of these there are what's called highly variable regions in the mitochondrial DNA where there are little mutations, and these are matches based on the location of the most distant pure female line ancestor. So if you know where your great-great-great-great-grandmother down the female line, you enter that into the database, and all of these matches, the green, the orange, the yellow, are different numbers of mutations separated from my mitochondrial DNA. This inset in here from this area around County Kalmar, or Kalmar County in Sweden, shows this white spot is where my great-great-grandmother, my Swedish great-great-grandmother was born, and the red matches are all of her exact mitochondrial DNA matches. So if I didn't know where my great-great-grandmother was born, I can get pretty close to it just by knowing her mitochondrial DNA matches. Now, it may be exceptional that all of these are zoomed in right near Kalmar in Sweden, but this is the result that I have. Okay, Y chromosome. this is going to get complicated. Only males have a Y chromosome, and it is inherited pretty much unchanged from your father. It does not undergo meiosis, so it doesn't change, it doesn't get scrambled with other DNA. And it mutates very slowly over many generations. There is an estimate, I saw an estimate that, let me explain a little more about this. There are exceptions to every rule, and I am one of them. Analysis of Y chromosomes, once you're pinned into a haplotype by your single nucleotide polymorphisms, or SNPs, then you're differentiated within that haplotype by things that are called short tandem repeats, which I've discussed before, which is stuttering in the gene, stuttering in the chromosome. The tests of known positions where STRs occur are numbered from they 1, 2, 3, 4, 5, 6, 7. They go up into the hundreds of beliefs locations. And the most distinctive and most variable are the first 12, up the first 25, the first 32, the first 64, and it goes up to 700 positions that can be tested for the STRs. The tests run between $100 and $400, and in theory, every male line descended from one generation should have almost identical Y DNA, except for me. All right, I jokingly accused my father of standing too close to the fluoroscope before my conception. He was an orthopedic surgeon. While I match other Newcombs of my lineage on the haplogroup, my STRs are very different. There is on the order of a 0.03% chance of any given STR to mutate in one generation. And I have approximately 100 generations of mutations from my nearest known Newcomb, which is a sixth cousin who tested his Y chromosome. And I have been unable to gain the cooperation of my only living first cousin, my known second cousins. I don't have any male line third cousins, and my fourth cousin. And I could test my brother, but okay, if it comes out that he's different, yeah, I know it was just me. But if he comes out the same, we haven't answered the question of where did the change occur, where did the mutations occur? And I'm not sure why it's important. But the same discussion of who tests, who resists testing, and the non-scientific, non-random selection of DNA testees is important to keep in mind when you're doing an analysis of your family and your genealogy and things like that using DNA. Okay, haplogroups. There's a classification scheme for the mitochondrial DNA and the Y DNA that pins down exact patterns of the DNA that classifies this is this group, and it's a tree structure, and it's very complicated. The naming convention is very difficult to understand. It started off when they started this off of A1, C4, and they would alternate numbers and letters going out every time they found a difference. Now they name it based on the defining SNP. So it's a complex tree structure that's difficult to understand. There are approximately 770,000 Y chromosome haplogroups. The tree is immense. It's something when you load it on your computer, you better have high-speed internet. The mitochondrial DNA has 258,000 haplogroups. My mitochondrial haplogroup is H2A1, it's more specifically H2A1BK. And my Y haplogroup is, when I tested, I had been RM269. And that's like, you have a tree and you've got the R group and you've got the M269 group that's out here on the limb. But I upgraded my test to 111 so they were able to be more specific and say I'm RS3251. But one of my Newcomb DNA cousins paid for the big Y, the 700 STR test, which nailed him down as an RBY 76323. And that's probably what I am if I were to pay the extra money for the $400 to get the more detailed test. But it's like going out a tree from the root to the branch to the limb to finally to the leaf. And this group here has just a very few members that have been identified in it, because you have to pay to have that test done. And it gains very little information for someone who matches very, very few people. I am an administrator for 3 Y chromosome studies on family tree DNA. I have at the lowest level at Y12, I have 18 matches on the Y chromosome. There's a British Newcomb who has thousands of matches. And why? I don't know. I'll go through these quickly. This was the origin of my talk. DNA in the Sons of the American Revolution. DNA can be included as supporting evidence, but there has to be other conventional paper trail to back it up. Only one DNA proof is allowed per application, and it can only be used to support a single relationship. That relationship has to be the applicant's father or grandfather. Evaluation only by a certified genetic genealogist and at the applicant's expense. This is the details. You don't need to know that unless you're filing an application for the Sons of the American Revolution. I assume the DAR has something similar. Okay, this is one of the points that I have been trying to make throughout my talk. Different groups test at different rates. Not all groups test at the same rate. In my experience, recent immigrant families test at higher rates. Families with extensive paper trail don't test. My wife is a quarter Basque. She has 784 pages of matches on Ancestry.com, on DNA matches on Ancestry.com. 25 of those matches can be identified as relating to her Spanish Basque ancestry. So that Basques are interesting. They don't seem to be interested in genealogy. They know they're Basque. That's all they need to know. They don't do DNA tests. And in France, it is illegal to sell a DNA test for genealogy purposes. It somehow or other violates the French citizenship paternity laws and that sort of thing. So they have some strange laws in France. It's not illegal to do a test, you just can't buy it in France. Swedes test at a high rate, probably because their naming convention is so confusing. Anybody who's done Swedish genealogy run into so many, you know, Lars Jacobsons and Anders Larsson, and it changes every generation. So they use DNA to help them sort out their ancestry. The Irish are in the medium range. Don't make any generalizations about blacks. Some of them won't get near a DNA test, and others are all into it. Don't make any generalizations about blacks and African Americans. Okay, DNA testing. Do you want a DNA test? Are you interested in DNA testing? What DNA test should you get? Well, the first question is, how good is your paper trail? There is really one thing that I found through DNA testing. I located a German cousin who had some more paper trail, you know, and that's basically the primary benefit I've gotten out of by testing my DNA. And so he was able to put me in touch with someone who had more information about my family, and I went back from the 1700s to the 1500s. Are you just curious? Do you need to know if you need to buy a kilt or lederhosen, which is an ad that Ancestry had that one time that just wound me up like nobody's business, because this guy thought he was Scotch and he had a kilt and all this stuff, and he turned out he was German, so now he has to buy a lederhosen. Are you an adoptee looking for your parents, looking for your father or your mother? Do you have any skeletons in your closet? You know, before I had did a DNA test, I checked with all my siblings and said, is there any reason why I shouldn't have a DNA test? Are you a sperm or egg donor at any point in your life? Are you concerned that your offspring through this process might be looking for you? Are you the last male in your family line? Gosh, I would love to have gotten a Y DNA sample from Curtis Chillison before he died. The Wattersons, who are now extinct in the male line, is a family I've studied, and they're gone. It is possible to do a DNA test and get your ethnicity estimate and not participate in matching with other people, so just keep that in mind. Here are some recommendations. If you're interested in autosomal matching, ethnicity estimates, that sort of stuff, Ancestry.com, buy the kit on sale for $40, buy it through Amazon, you don't pay the $10 shipping. They have the largest database. They do a bang-up job on autosomal DNA. Y chromosome or mitochondrial DNA, you do FTDNA, it's $100 to $500. Health, my recommendation there had been 23andMe. 23andMe is going bankrupt, so I don't know what to recommend for health right now. I will tell you that if you go to FTDNA, they have a health option, but if you want to download your raw DNA data, your raw autosomal data, they edit out the red flag, the health red flags. They null them out before at least they used to, before before you download, because they don't want you making medical decisions based on their DNA analysis. They think that's basically something for doctors. Okay, do you have your great-grandmother's toothbrush? Or like I have a lock of my great-great-grandmother's hair. Do you want it analyzed? This place called To the Letter DNA will attempt to extract DNA and charge you a fee for that. They then analyze whether the DNA is viable or not, and they charge you a fee for that. It's a stepwise process. The actual analysis of the DNA is up to $2,000 for, you know, if you have somebody's baby tooth or something like that. They give you, they do a full a full genome sequence. They figure if you're going to be paying that much money, you might as well do a full genome sequence. The question, of course, is what do you do with it after that? Because some matching services, Ancestry does not accept uploads. 23andMe does not accept uploads. Family Tree DNA accepts uploads. GEDmatch accepts uploads. Some of the services will and some of them won't. Some of them are changing their policies pretty much as we speak. If you're an enthusiast, you will download your raw DNA file and then upload it to GEDmatch. And GEDmatch is a service which allows people who have tested with different companies to put their data on one database and match it and use some advanced techniques. And GEDmatch allows you to look at other people's DNA besides those that just match you. So if you know someone's handle, his DNA identifier, you can match it, and they have these, they're the ones that host these services, these tools such as Lazarus, which I spoke about before, which allows you to build a virtual DNA of your ancestor. And as I've mentioned before, you can download your DNA from any of the services that do DNA testing, and they all allow autosomal DNA download, and some allow upload from other sources. GEDmatch is a freemium product. They allow basic access and basic matches for free, but their higher level of service is actually rather expensive, and it has the advanced analysis tools. Ancestry has recently introduced what they call Pro Tools, which is an extra cost-based service, and you should have some concern for privacy. What someone can do with your DNA data, I don't know. Most of these services, you know, you're in a world where your accounts are being hacked and your personal information is out there. What can someone do with your DNA data? I can't say for sure, but you know, people. I've heard that you know, like the Chinese were trying to do a DNA analysis of Americans to find some disease that would only affect Americans, you know, and how that would. But I have one cousin who's a bit of a bunker, you know, dweller. And he flat refused to do a DNA test at my request. He's my first cousin, my only living male line first cousin. That's the end of my talk. Now, do I have questions?
Okay.
Right, and I should have prefaced this all, if I have any genetic genealogists or lab technologists or biochemists, you know, please speak up and correct me. I've been finding errors in this talk, you know, since I wrote it the first time.
I had my mother and dad's DNA done by Ancestry before they passed, and then I just did mine on MyHeritage. I have got from all over the world. And theirs is just basically US. Is it just because of their clientele, or?
This is another important thing that I didn't mention, I forgot to mention. Ancestry is primarily US, and a lot of foreigners use MyHeritage.
Okay.
And you can download your parents' DNA, raw DNA from Ancestry, and upload it, at least you used to be able to, and upload it to MyHeritage.
Mm-hmm.
And there are, because there are certain differences in the tests that are performed and the SNPs that are provided in the upload and the downloaded data, they don't guarantee, or they don't suggest you take their ethnicity estimates very seriously, but their matches are still valid.
I have one match from my parents on Ancestry and one on MyHeritage.
And you can also download your DNA and their DNA, and upload it to GEDmatch. And in GEDmatch, you will tend to find people. In Ancestry, you'll find somebody who matches who you match, who has no tree, and you will attempt to reach out to them and contact them and say you were DNA matches. And
some of them will answer you, and I've gotten that. Well, I don't recognize that name, and that's it.
Right, okay. And in GEDmatch, you tend to get people who are more studied up on DNA, genetic genealogy, and are probably more willing to correspond with you. They're probably looking for some contact. Okay.
When
Yes.
is the follow-along course? I'm saying this because before we started, I reminded him that I'm here for a review, and I learned more today than I did the last
Well,
time.
you have to understand that today took approximately 1 hour, whereas three minutes per slide, whereas at the SAR meeting, I was rushing through it at 1.5 minutes per slide to get in under the 30-minute time limit. But you need to, I'm a case of a person who got interested in genetic genealogy just because it's a subject I was interested in studying, not because it was anything that was terribly important to me. So I've spent maybe $500–600 on DNA tests. And would I have been better off spending that on records? On paper trail stuff? Probably. But I gained a little more understanding and a little experience being able to.
Which is the best genealogical testing service for somebody who has Caribbean ancestry? People who came from France and Spain and then came through the Caribbean to New Orleans?
Well, you see, the thing is that you want to get the most bang for the buck. And if you don't have specific questions about your pure maternal line, about your mitochondrial DNA, and you don't have specific questions about the paternal line, let's say through your brother or something like that, then Ancestry has the biggest database, but Ancestry doesn't allow uploading data. So for $39, $40, you can do an Ancestry DNA test, then download the raw DNA data, and as of the last time I checked, upload it to MyHeritage. And that gets you and you can upload it to Family Tree DNA. And that gets you 2 or 3 exposures for the price of 1. And so I know that there are services that claim to be able to place you in a particular Indian tribe based on your DNA, and most of these are considered to be bogus. It's considered to be, but I don't know of any specific test for Caribbean ancestry. The Caribbean is a big mixing pot with just, you got French, Danish, British, African, Indian, East Indian, Caribbean. So I don't think I have an answer for that question, although I do have a plan to get you the most exposure for the money. Okay?